Hello, everyone. Good afternoon, and welcome to the latest edition of Brain Bridge Talks by CDM. My name is Anushka, and I'm a science communicator here at the Center for Brain and Mind. And this webinar is one of our many efforts to reach out to people who are interested in our work and to learn more about the cutting edge research that we're doing in trying to understand the molecular and genetic basis of psychiatric illnesses. I'm just going to go over some quick rules for the webinar, and then I will be handing it over to my colleague, Dr. Ashita, who will handle the introduction and the Q&A session for today's webinar. As always, please remember to keep yourselves muted. You can keep your cameras on if you'd like, but please do not unmute yourself during the webinar. We will allow you to unmute yourself for the Q&A session in case you want to ask your question in person. If you have any questions for Professor Srivastava, you can leave them in the chat box and we'll get to it in the last 15 minutes of today's session, which is the Q&A. For those of you who have to leave in the middle, or if you have any colleagues who can't join today, we will be recording this session and putting it up on our YouTube channel in a few days, and you'll get an email with the link once that's done. If you have any other questions, we'll also be leaving Professor Srivastava's contact information, so you can reach out to him if you have any further questions. So without further ado, I'm going to hand it over to my colleague and we'll begin today's session.
Hello, everybody. Good afternoon. I'm Dr. Ashita. I'm Inspire faculty in the Department of Psychiatry, and I'm an investigator at the Center for Brain and Mind. So this afternoon, I'm very happy and delighted to welcome my collaborator and mentor, Dr. Deepak Srivastava, group leader at the MRC Centre for Neurodevelopmental Disorder at King's College London for today's edition of the Brain Bridge Talks. Dr. Srivastava leads a pioneering research program investigating how synapses are regulated in health and disrupted in neurodevelopmental and psychiatric disorder. As the director of the World Cellular Imaging Center, he integrates advanced cellular imaging, molecular approaches, and patient-derived stem cell model to uncover how genetic risk factors shape synaptic biology and neural circuitry. His work spans fundamental mechanism of brain connectivity to translational questions relevant to conditions such as schizophrenia, autism spectrum disorders, and Alzheimer's disease. Through close collaboration with clinicians and industry partners, his research aims not only to deepen our understanding of the brain function, but also to inform future therapeutic strategies. We look forward to an engaging and thought-provoking session with you, Doctor. I would like you to please take over the stage.
Thank you very much for that very kind introduction. It is a real pleasure to be here and to be part of this seminar series. I was just saying, and I would like to thank both Ashita as well as Anushka for organizing this here and inviting me. And I was just saying just before the meeting that I've had the pleasure of coming to visit NIMHANS, and I've met a few of you before previously, and I have very fond memories of visiting and seeing the amazing complex, the hospital, as well as the academic portion, as well, and labs. I think it's a truly amazing and unique place in the world. So it's a real honor to be able to speak today. I've also seen that on the in the audience, we have Professor Raghu Pinanjat. So when I was a PhD student, Professor Pinanjat was a group leader where I was doing my PhD at the Babraham Institute in Cambridge. So I somewhat feel as if I've gone back in time a little bit and feel a little bit anxious, like a PhD student presenting to a senior colleague. But the familiarity here is fantastic. So what I would like to do today is to tell you all a little bit about some of the work that my lab has been doing, where we particularly have been trying to use human-based cellular models to study the origins of schizophrenia. And I would like to tell you a little bit about work that we've done where we've been focusing on a specific gene, a gene called ZNF804A, which will hopefully become apparent why we coined this here as the original common variant. So the work that I'm going to speak about is not work that I've done personally, but work that has been done by a group of people in the lab. So I'm incredibly fortunate to work with a really engaging and exciting group of early career researchers. And the work that I will be particularly talking about is really being driven by this person here, Laura Sicklinger. She is a former PhD student. She's now a postdoc at UPenn in the US. And she's the main driver of this work that I will speak about. She collaborated a lot with other members of our group, as well as being co-supervised not only by myself, but by my close friend, Tony Vernon. Professor Vernon's lab and my lab work very closely together. And we also collaborated with other people around the world, including this gentleman here, Max, who was a PhD student at the University of Bochum and is a proteomics expert. So I think this is probably one of the most important slides I have, and that is really to thank the people who have done the work. So the work that my lab is really interested, the kind of global question we have is to really comes from this idea of trying to understand what are the molecular mechanisms that underlie the communication of information within the mammalian cortex. We're interested in understanding what are the intrinsic as well as the extrinsic factors that drives this or underlies this communication of information, and also how this occurs both in health and in disease. And although I will not talk about this here today, we're also very interested in how these effects or these mechanisms may differ in the biological male or the biological female brain. So realistically, we're very interested in the whole lifetime of the cell. So we're very specifically are interested in excitatory neurons that are found within the mammalian cortex, particularly the mammalian forebrain. We're interested in how neural stem cells know what type of neuron they should become and where they should migrate to or locate themselves within the developing cortex, how they develop their beautiful dendritic architecture, and then also how then this changes into how the site for where the majority of excitatory synapses occur, dendritic spines, how they're formed, how they're maintained, and how they're remodeled. And in conjunction with this here, we even are interested in how the thousands of proteins that are found at synapses are organized and how they are dynamically regulated. And we ask these sorts of questions, not only in the context of how this occurs under physiology, but how these mechanisms may be altered in a variety of different disorders or conditions, primarily focusing on psychiatric or neurodevelopmental disorders. And one of the ideas that we are really trying to explore is what could be the pathogenic mechanisms that underlie the appearance of aberrant, either neuronal morphology or synaptic morphology, that could ultimately contribute to altered neuronal circuitry, which may underlie the altered psychological, clinical, and cognitive deficits that are associated with various different neurodevelopmental and psychiatric disorders. In order to do this here, as I've already mentioned, we really are focusing on using human-induced pluripotent stem cells. And by way of just a very brief introduction, this is the technology that was developed by Professor Shinya Yamanaka, that was published back in 2007, where he demonstrated it was possible to take an adult somatic cells, such as skin fibroblast, blood, or hair keratinocytes, and using for genetic factors, the Yamanaka factors, reprogram these cell-defined somatic cells back in time to a pluripotent state. So these cells have the potential to go on and develop into almost any type of cell that's found within the body if exposed to the right environmental conditions. And we've been really using this technology to be able to generate human neural cells that we can grow in a dish. We're very lucky that we have access to a small biobank of patient-derived iPS cells, as well as iPS cells from apparently healthy individuals. Our biobank is predominantly defined by diagnosis first selection of participants. So participants with a particular diagnosis, such as schizophrenia or autism. And in certain cases, we also have individuals which have been identified by a gene-first approach. So they're known to carry particular copy number variants or deletions, mutations in specific genes. We also are embracing gene editing approaches, which allow us to either introduce, correct, or manipulate genes within our iPS cells. And we're typically generating either 2D cultures, generating neuroprogenitor cells, neurons, or organoids in our lab. The work we do with glial cells is predominantly with our close collaborator, Professor Tony Vernon. But we're using these cellular platforms to ask a variety of different questions, everything from trying to understand underlying mechanisms that may result in an increased likelihood of somebody developing a psychiatric or neurodevelopmental disorder, to try and understand how environmental factors may influence neurodevelopment or interact with genetic alterations, and even as a platform to carry out studies to understand the mechanisms of actions of different drugs. So very simply, we use various different approaches and to generate our neuronal cultures. We use directed differentiation approaches where we use small molecules to guide our iPS cells through the different stages of neurodevelopment to really try and recapitulate the early stages of neurodevelopment, ultimately resulting in neurons. When we also use forward programming approaches where we're overexpressing transcription factors, such as, in this case, neurogenin 2, to generate a more pure population of glutamatergic neurons that acquire functional activity in a shorter amount of time. And we're also using different approaches to generate 3D neural organoids, which we can use in various different ways, either as whole organoids or as these air-liquid interface slice cultures, where we perform a variety of different assays. As she just mentioned at the start, we are very interested in using imaging-based approaches. We use both imaging of fixed samples as well as live cell imaging to really try and understand the morphology and architecture of our samples. And we're moving more and more into using more functional assays such as multi-electrode arrays, where we are particularly using them to understand the development of network properties. And very generally, we have several quite big research questions in the lab. I'll be speaking a little bit about our approach to trying to translating genetic risks into a neurobiology of neurodevelopmental psychiatric disorders. But we also have programs where we're looking at trying to understand sex differences in health and disease, as well as the potential roles of estrogens as a potential therapeutic targets, trying to understand the molecular mechanisms of drug action. A more fundamental question is trying to understand mechanisms of structural plasticity, and then also work that we're also collaborating with Dr. Shita with is understanding or investigating neuroimmune interactions. But what I'm going to do today is to really think about the work that we've been doing with schizophrenia, and I don't really need to introduce this disorder to yourselves, but just to really highlight a couple of things that, schizophrenia not only is it a chronic disorder, it is also incredibly heterogeneous. We know that the positive, negative, and cognitive symptoms are all very much involved in this disorder, and whereas antipsychotic treatments are posited to have a good impact on the majority of patients on the positive symptoms that they experience, there's very little in the way of treatments, particularly from pharmacological approaches to treating the negative or the cognitive symptoms, which is possibly quite important when we think about wanting to try and integrate patients back into society, as the severity of these negative or cognitive symptoms may be a key barrier for them. Now, in order to really tackle the heterogeneity and to try and create a better understanding of the underpinnings of schizophrenia, I would say close to almost 2 decades of work where high throughput genetic studies have really started to help us understand the complex etiology of schizophrenia. So in these studies, which include genome-wide association studies, as well as exome studies, and more now moving towards whole genome sequencing studies, have really allowed us to understand that the complex etiology of schizophrenia could almost be split into 2 sort of categories. On one hand, we have rare variants, or copy number variants, such as the 22q11.2 deletion, which is possibly the most penetrant copy number variant when it comes down to schizophrenia, as well as other factors or chromosome regions or genes that are thought to have high penetrance, but they do not occur very often within the populace. But then we also have, on the flip side, a host of maybe over 250, close to 300 common variants, which are located throughout the genome, which occur quite often in the populace, but have a low penetrance. So that's to say, if you have a variant in a particular gene or gene locus, that the odds of somebody developing schizophrenia is only increased by a small amount. What is interesting, though, is that many of the genes, so many of the variants, do not lie in coding regions, but those that do or have been identified to impact the expression of specific genes, they appear to result in a number of molecular and cellular disturbances, which can be broadly categorized as altered gene expression or pathway function. A range of biological processes are also thought to be altered, but very much and very often, either directly or indirectly, many of these common variants are thought to impact synaptic connectivity, synaptic function, and therefore potentially impact altered circuit function. Interestingly, in the most recent study of common variants from the Psychiatric Genetic Consortium, they made the observation that many of the genes that have been associated through genome-wide association studies and schizophrenia actually encode for genes that are synaptic, with a very much over representation in genes of proteins encoded by these genes found at pre and post synapses. And this is quite interesting when we think about human studies, where classic studies from David Lewis's group, for example, have shown that in post-mortem studies, there is a general trend to a reduction in the number of dendritic spines and specific populations of pyramidal neurons found in the cortex. but more excitingly, in recent studies where researchers have been using a PET ligand against the synaptic protein SV2A. So this UCBJ PET tracer have identified that in participants diagnosed with schizophrenia, there's a reduction in the amount of this PET tracer in various different brain regions, suggesting that there is an impact of diagnosis on synapses, i.e. schizophrenic individuals may have reduced synapses in the brain. Of course, these studies, even these PET studies, have a number of caveats, such as SV2A is not found at all synapses. It is also found at excitatory, inhibitory, and even neuromodulatory synapses. So we don't really know whether or not the impact of this reduction in SV2A of this PET ligand is telling us that there's a reduction in a particular type of synapse. And we also do not have a cellular or synapse level resolution here as well. But the real take home here is that multiple lines of evidence, both genetic as well as in vivo and postmortem studies, really kind of point to this idea that synapses are affected in a disorder like schizophrenia. But how are they affected? What are the molecular mechanisms that may be driving this? And at what point during the life course of an individual who does go on to develop schizophrenia can these alterations in synapses be identified? So when does this pathogenic mechanism start? So about just over a decade ago, my lab started to get very interested in this gene, ZNF804A. And that was because the first variant to reach genome-wide association for schizophrenia back in around 2008 was a variant that was found within this gene, ZNF804A. And the variants have been continuously identified in this gene, and it is now considered to be quite a robust risk variant for schizophrenia. So we call it the original common risk gene because variants of this gene were the first to be identified. And if we think about the human studies have examined individuals who carry this risk variant, there are some neuroimaging and neurocognitive phenotypes that can be identified, everything from reduced interhemisphere connectivity to decreased activity, which broadly speaking have been associated with altered executive functioning, cognitive memory, and social cognition. So this here kind of fits in nicely that this gene is having an important role within the human brain, and that variants in the gene could result in alterations or contribute to alterations in domains that are relevant for schizophrenia. So as I said, there are multiple variants in the gene that have been associated, some in different populations. It's not just Caucasian population, which is good to in some ways to see, but the one that is most robustly identified, or the first one that was identified, is this RS1344706, which actually lies within intron 2 of this gene. So the gene itself is comprised of 4 exons. It is predicted to generate multiple different variants, but the 2 variants that are particularly of interest are the full-length protein as well as this E3, E4 variant. What's interesting is that we know the gene, the protein itself is quite a disorganized protein. It doesn't have very many domains. What it does have is robustly this this zinc finger C2H2 domain, which is found on the N-terminal region, and two nuclear localizing signals, which are found midway down the protein as well. And it was from these observations that the initial idea was that ZNF804A was indeed a transcription factor. And indeed, when the original functional studies were done, they involved knocking down ZNF804A in a variety of different cell types, and genes that were associated with cell adhesion, migration, cytokine signaling, and neuronal morphology were identified. So this is where we started our original work here, thinking that, well, this is not only a robust gene associated with schizophrenia, it appears to have a role in controlling the expression or potentially the function of cell adhesion, neuronal morphology. And so let's try and model what it's actually doing in human neurons. And one of the first things that we did was we took a conditionally immortalized human neural progenitor cell line, and we just looked at the where ZNF804A was expressed within either neuroprogenitor cells or the neuroprogenitor cells that we had differentiated into neurons. And the first thing that we saw, which really was quite surprising to us, was that ZNF804A was not just localized within the nucleus, but also out in the cytoplasm. And indeed, if we looked at the neurons, we could see that it wasn't just in the cell body, but along MAP2 positive neurites. Indeed, if you overexpress a GFP tagged version of the protein, we can see here in HEC293 cells that near the extremities of this cell, actually you can see ZNF804A localized quite clearly. We went on to demonstrate that this sort of unexpected localization of ZNF804A could be seen in various different cell types. So we took human iPS cells and derived neurons, and we could see, again, yes, it was expressed in the nucleus, but along the MAP2 positive neurites, and in fact, it seemed to co-localize with a number of postsynaptic proteins, such as PSC95, as well as the GLUN1 subunit of NMDA receptors. Moreover, when we used primary rat cortical neurons to derive neurons that had a much more mature morphology, we again saw a similar pattern with ZNF804A or its homologue, ZFP804A, being expressed along neurites and specifically within dendritic spines. Indeed, when we took a more of a functional approach where we used the short interfering RNA molecules to knock down ZNF804A in these primary rat cortical neurons, we found that there was a reduction in the number of dendritic spines. And moreover, when we challenged these cells with a chemical LTP paradigm, which ordinarily would increase the number of dendritic spines and result in an increase in the size of dendritic spines, we found that whilst we were still able to increase the number of dendritic spines in cells with reduced ZNF804A expression, these dendritic spines did not show an increase in size. So we saw that there was an alteration in the ability of these cells to respond to structural plasticity stimuli. And this is being somewhat replicated by other groups where they have taken approaches to, say, for example, overexpress different forms of ZNF804A, and they have found alterations in the density of dendritic spines in these cell types as well. But one of the problems here was that we had intervened or altered the expression of ZNF804A in those experiments after the cell had already developed. And when Laura came to the lab, she asked this question, quite a simple question of when is ZNF804A most highly expressed? So she looked at the data from the Allen Institute's brain span data set. She looked at data from 8 weeks post-conception to 40 years of age from human tissue, the various different parts of the cortex. And what she found was that ZNF804A was most highly expressed at this early stage of development. So this early mid-prenatal stage of development is where we saw its highest expression. And indeed, this is consistent with some observations from Professor Nick Bray's group at Cardiff University, which suggested that the variant of ZNF804A actually reduces the expression of ZNF804A, particularly within this early mid-prenatal development stage. So Laura went on to try and understand which cell type was expressing ZNF804A, and again, using a data set which was derived from the developing fetal cortex using single nuclei RNA sequencing approaches. What she was able to find was ZNF804A was most highly expressed in developing excitatory neurons, and actually showed the lowest expression in microglia. So this really led us to hypothesize that not only is early development a key point for when ZNF804A may be exerting its function, but that the developing excitatory neurons was the primary cell type. Thus, if we were to want to try and understand how ZNF804A could contribute to the development of schizophrenia, that looking at developing excitatory neurons would be the cell type to look at. In order to do this, we decided to use iPS cells and in combination with this forward programming approach, so this overexpression of neurogenin 2. This was because, well, within 7 days, we can generate glutamatergic neurons, shown here by the expression of MAP2, as well as VGLUT1. And we can keep these cells in culture for long term, up 20, 30, 40 days plus, and that these cells also rapidly produce electrically active cells after about 2 weeks to 3 weeks in culture. So what Laura also did was to not only use this forward programming approach, she also combined it with a small molecule patterning as well to really try and lock the cells towards a forebrain fate. And here's just a demonstration that the cells after 7 days not only are positive for VGLUT1, but they're also positive for the upper layer cortical marker CUX2. I don't show this here, but they are not positive for deep layer markers such as CTIP2 or TBR1. Most importantly, we can see that within 7 days, there's a very large increase in the expression of the transcript for both the full length as well as the E3, E4 variant of ZNF804A. And we can see that it is nicely expressed along GFP positive neurites of these cells as well. Now, to really try and understand the function of ZNF804A, what Laura did was to take a functional genomics approach and generated an isogenic heterozygous and homozygous mutation lines. She did this by using a dual guide CRISPR approach. So she used guides that were sat either side of exon 3 and excised exon 3 to generate a heterozygous as well as a homozygous mutant line. We also generated wild type cells. So these are cells that were that underwent the whole genome editing process, but had not actually taken up this deletion. So we had 2 clones of each genotype, which were fully characterized, and we confirmed the presence of this deletion or not. And what we can find is that when we look at the expression of ZNF804A, that we see a gene dose-dependent reduction in the expression of the protein, with the hets showing roughly about 50% expression compared to the wild type, and the homozygous showing a much greater reduction again. So we were confident that we now had a nice isogenic mutant loss of function line that we could use to try and understand ZNF804A's function in these developing excitatory neurons. One of the first things we did was to carry out an RNA sequencing experiment, and we just really wanted to do a bit of a sense check to make sure that we were not altering the fate acquisition of these cells by mutating ZNF804A. And here we just looked at the expression levels of various different genes, everything from pan-neuronal to genes associated with upper layer or deep layer fates. And here we have the wild type samples, the heterozygous samples, and the homozygous samples. And you can see from this heat map that they're broadly almost identical in every way you look at it. So we're not altering the fate of these cells, which I think is an important question to ask when generating such a system. When we look more closely at the whole transcriptome and looked for differentially expressed genes, we actually found that there was quite a subtle impact of ZNF804A. And this is just demonstrating the differences in the genes that are showing differential expression between wild type and the homozygous cells. So we're only seeing close to 50 genes showing either increased or decreased expression. What's interesting is that if you take these 50-odd genes and look at what they were enriched for, we can see an enrichment for genes involved in cell adhesion, which is very much like what we've previous studies had seen, but also for genes that are associated with synapses, and particularly post-synaptic genes. Interestingly, we also could see that some of the genes, particularly the genes that are downregulated following ZNF804A or loss of ZNF804A, which are also seen to be altered in expression in the brains of individuals with schizophrenia. So there's some overlap with schizophrenia differentially expressed genes. Because of the association with synapses, we designed an experiment where we could carry out a high content imaging approach to really look at synapses, not just at individual cells, but where we could look at hundreds, if not more, genes, cells within our cultures. So these are an example of some of the images that we could take. So we would take from 96 well plate. Each well we would take between 10 to 15 fields of view. This is just one field of view where you can see, if we zoom in to this bit of dendrite, this is what the MAP2 looks like. This is what the PSC95 looks like. And here we've got another example of what the V-GLUT1 staining looks like along MAP2 positive dendrites. And we carried this out using various different combinations. So we looked at both pre and post synaptic overlap using a combination of PSC95 and V-GLUT1 or GLUN1 and SV2A. And what was really surprising to us was that we were actually seeing an increase in the number of putative synapses. So this is just the number of overlapping PSC95 V-GLUT1 or GLUN1 SV2A synapses. This was not expected at all. And we've gone on to carry out some functional experiments to see if this is actually changing the excitability of the cell. So we've used calcium imaging, and we've evoked a calcium response by applying potassium chloride to the cells. So we're not looking at synaptic transmission per se, but just looking at the excitability of the cells in many ways. And what we found was that in the homozygous cells, we saw a much greater response to this potassium chloride stimulation. So what this here really kind of tells us is that in these developing neurons, it appears that ZNF804A a mutation seems to result in an increase in a putative synapse formation, which is coupled with an increase in the excitability of the cell. So the last thing we really wanted to ask was what could be driving these effects, and to really try and understand how ZNF804A is exerting their effects in the cells. We really hypothesized here that, well, ZNF804A doesn't seem to be a major driver of regulating gene expression, despite what the purported functions of this gene were. And when we looked at the literature, a number of these two hybrid based studies had actually identified that ZNF804A could interact directly with a range of different proteins, including cell adhesion or cytoskeletal proteins, but very interestingly, also proteins involved in translation or RNA binding. These are from different studies, and this is a very interesting observation. So, and it was in fact actually shown in this study that ZNF804A is a regulator of protein translation. So we hypothesized that this may be the case as well in our developing neurons. And we wanted to take an unbiased approach to asking this question. And we specifically wanted to look at the proteome of our cells, the wild-type heterozygous or homozygous cells. And we specifically wanted to look at the proteome of the cell, the soma, versus the neurites, to really try and understand if there were alterations in the proteome in this subcellular kind of regions of the cell. So we did this, or Laura did this here by growing or differentiating her iPS cells into glutamatergic neurons by growing them on this mesh. And here we just have a confocal image where we can see the mesh here. In blue are the DAPI nuclei, and in magenta are actually the neurites, which are dangling down. She then separated the cell bodies and the neurites and sent them for proteomic sequencing with our colleague Max and Bochum. And this is just to try and convince you that we could nicely separate the soma from the neurites. This principal component graph here shows that all the samples that were the soma separate very nicely from the neurite samples. And if you look at the proteins expressed in the soma, they are very much enriched for proteins that are involved or found in the nucleus, histone proteins, nucleosome proteins. And in the neurite, they're very much in proteins associated with cytoskeletal cell membrane actin binding. So we got good separation. And then when we look at, so in this case, specifically the neurite compartment of our wild type versus our het or homozygous mutant neurons, we found that very interestingly, these terms translational or ribosomal proteins were particularly appearing. And what we were finding was that there was this alteration in the distribution of these ribosomal proteins, which we confirmed by immunohistochemistry. Here we're just showing that this ribosomal protein S6 shows an increase in abundance along MAT2 positive neurites in the mutant ZNF804A derived neurons. So we hypothesized further that this may be a reflection that there is an alteration in local protein synthesis within these cells. And indeed, if you looked at protein synthesis through a proxy of phosphorylation of this ribosomal protein S6, we could see that the het and the homozygous mutant lines show an increase in the phosphorylation. And more importantly, when we use the sunset assay by basically by incorporating puromycin and visualizing this here in our wild type or mutant cell lines, we could see in our heterozygous or homozygous cell lines there was a greater incorporation of puromycin compared to the wild type cells, specifically along the MAP2 positive neurons. So with that, what I would like to summarize very quickly that based on the data that we have observed in our two studies, we believe that ZNF804A does have a role in regulating gene expression, particularly of cell adhesion proteins, but that it also has a function in regulating protein translation as well as synaptic morphology, both in the maintenance of synaptic morphology, but also in the formation of synapses. And that these effects seem to be driven in part by the ability of ZNF804A to regulate the expression or the abundance of ribosomal proteins along dendrites, which then have an impact on the expression of synaptic proteins and potentially consequently on the number of synaptic proteins, synapses being formed. So with that, again, once more, I would like to thank the people who have done the work. We have a great lab. We have amazing collaborators. And I'd also like to thank the people who have given us money to actually do this work. And thank you all very much for your attention. And I'll be very happy to take any questions that you may have. Thank you.
Thank you very much, Dr. Deepak, for this wonderful, stimulating conversation. From my understanding, it's harder to work and understand the role of common variants and specifically tease out their molecular function in disease context. So it is a very, very motivating and stimulating presentation. Thank you for that.
Thank you.
We request questions from the audience. So, sir, could you please stop sharing your screen so that...
Absolutely. There we go.
Yes. So, Kasturi Saha her question is whether you also focus on dissecting the precise functional effect of individual risk variants beyond analyzing Jiva's risk loci. I'm also curious about your thought process. When prioritizing common variants for experimental validation, since many Jivas associated variants may be proxies rather than truly causal. And the nearest gene is not always the effector gene. How do you decide which variant or loci are the most worthwhile to study experimentally?
So Katsuri, that is, I think, two really really important questions. So the first, I will say this, I don't think I have a very good answer for either of them, but I'll do my best. What I would say is, in the first one, how do we... So one thing that we have taken, the approach that we have taken has been really to try and understand the function of the gene. So we're not studying the actual variant per se, in the case of ZNF804A. We've gone for a bit more of a brute force approach and just try to disrupt the whole gene rather than trying to precisely introduce the variant to understand what that variant is doing. And one of the reasons we have done this here is that, for example, work that has been looking at the impact of the variant in terms of gene expression suggests that it may only reduce ZNF804A's expression by 5 to 10%. So by itself, that variant may not induce very strong cellular phenotypes. So the question we've really just been focusing on is, what is the function of the gene, rather than trying to model the actual gene itself. And the second point about you said about gene prioritization, I mean, quite simply, in my opinion, you need to work closely with true geneticists, thinking about EQTLs, thinking about maybe even going further to think about, regulatory sites, epigenetic regulatory sites, and long-range cis and trans interactions as well. So it's not something that I think is very easy to address, and it's certainly not something that my group can do directly. But I think it is interesting, and what I would also say is that we again have focused on genes where the variants are clearly within a gene, but then also try to think about how do they interact with other genes that have are also associated with common variants. So, like I said, not a great answer, but I think the best I can give you right now anyway. So.
Thank you for that. So Nivedita asks, if length of dendritic spine influence the connections in the brain, and do you find any correlation like shorter dendritic spine, lesser cognitive ability?
So we haven't done this work directly in this study, but there are a number of studies, some from our group, actually some from my postdoctoral research, and I think a lot more work now coming out, which tells us a little bit more about the nuances of looking at dendritic spine morphology and thinking about it in terms of cognitive ability. I mean, classically, we think the number of dendritic spines is important, but we also know that the morphology of them are also equally important. It's a little bit context dependent, but I would say that there are some great studies out there that have tried to answer that question a little bit more. In the context of what we've done, what's an interesting observation, I would say, is that the loss of ZNF804A may result in the loss of the dendritic spine specialization, but not necessarily the synapse itself. So if we look at the number of pre and post synaptic proteins, what we're actually seeing is, rather than having in the primary neuronal cultures, rather than having a presynaptic synapse onto a dendritic spine, it seems to be a presynaptic synapse that seems to be directly on the dendritic shaft. So we've lost the structure, but we haven't lost the synapse. So I hope that answers your question a little bit.
Kasturi has another question. The soma proteins seem closely linked to chromatin remodeling. Has there been any work, or are there plans to explore the role of chromatin remodelers more directly in schizophrenia?
Yeah, absolutely. So, I mean, I think there's two great examples. There's studies that have been done looking at the gene MEF2C or another gene SETD1A. These are both transcription factors or chromatin remodelers, and they definitely are of interest to a lot of groups, but not something we've worked with directly.
The name is not mentioned here, but there's another question. Is there evidence that adverse childhood experiences like bullying moderate the neurodevelopmental impact of ZNF804A specified variants?
I do not think that there's been a direct link with bullying, but there are suggestions that there may be an interaction with early life stress, um, potentially maternal immune activations. So when the child's in utero, there may be some interaction with an increased cytokine profile or release. Um, um, but I don't think bullying directly. But there's definitely some interactions and interesting interactions to look at in terms of other environmental, particularly early life stress, I think, to be explored. So, yes.
Next, Dr. Raghu has a question for you. Dr. Raghu, could you please unmute yourself and ask the question directly?
Yeah, hi Deepak. Nice to see you again. A very nice seminar.
Thank you.
I had a couple of questions. The first one is the following, which is that your essential observation from the proteomic study was evidence of increased protein synthesis. Has that been seen in schizophrenia-derived iPSC lines?
Yeah, so that's really interesting. So there's actually a few studies that have kind of suggested this here, that there may be alterations in at least global protein synthesis.
Want an OTP?
Yeah, I mean, I think one interesting study is from Kristen Brennan's group. It's actually not such a one of her big papers, but I think in her classic cohort, she looked at global protein synthesis in the neuroprogenitor cells as well as in the neurons, and saw that there was an increase in protein synthesis. She didn't go into any more of the mechanisms and whether it was actually increased local protein synthesis, i.e. in the absence of gene transcription. But I think there is some precedence for this here. So I hope that answers your question, Raghu.
He mentioned he has a couple of questions, so I'm waiting. Dr. Raghu, please go ahead. Okay, so there are more questions in the chat box. So Balraj is asking, is there a way to understand one's case of schizophrenia? I was diagnosed with schizophrenia affective disorder, but I had very little knowledge of my illness. I understand this question might be a bit out of scope with today's webinar, but any suggestion or advice in general might be helpful.
Well, thank you very much for that question. And I would first of all say that I myself am not a medical doctor or a psychiatrist. I am a fundamental researcher. So unfortunately, I may not be the best person to speak about in terms of the clinical side. But in terms of trying to understand, the molecular and cellular side of things, there's quite a few resources online that try to bring together the information that we have about this disorder. One website that I could suggest is the Brain Behavior Research Foundation. They have excellent information that span everything from what may be the underpinnings to the clinical course. And another really good resource is a foundation here in the UK called MQ Mental Health. They also have a lot of very good information, and they also have links and contacts to patient and carer groups as well, which may be an interesting area for you to explore. So I hope that helps a little.
Thank you. Dr. Raghu, could you please ask your question?
Yeah, Deepak, the other question I had was probably a difficult one, but it's still unclear what is the function of this protein as a molecule.
Yeah. So I think that is it's really good. So if you look at the, I have this exact same question all the time, and I think if we look at the interactome, and that has come from a number of different studies, it's actually quite narrow. It's not that there seems to be a lot of proteins that it's interacting with. But two observations that I have come to is that one, there is a lot of evidence that ZNF is somehow interacting or regulating adhesion. Is it an adapter protein directly interacting with adhesion proteins? The yeast-2 hybrid studies would suggest, yes, this is the case. With other interactors, I think, how is it? I mean, we're thinking about it in terms of protein synthesis. What is it doing here? Is it binding RNA? Is it directly interacting with the ribosomes and controlling its localization? Honestly, Raghu, I don't have a good answer for that. I have lots more questions, but not good answers at all. So, yeah, sorry, not a very fruitful answer.
Thank you.
Thank you, Raghu.
Thank you, Dr. Raghu. So DP and Nikita, both of them have a question. Does ZNF84A influence both schizophrenia and bipolar disorder, or is there a shared common molecular pathway?
So actually, very interestingly, I would say that it is originally ZNF8048, or this variant in particular, the original one was associated with psychosis more broadly. So it was both schizophrenia and bipolar. What's really interesting is that as we've gone on and the genetic studies have become larger, there's still evidence that ZNF is associated with bipolar, but there's also, it appears that ZNF may be more, the variants are more associated with schizophrenic individuals with negative symptoms or strong negative symptoms. Beyond that, I couldn't really tell you much more, but I do believe that there is a strong link with bipolar. So could we be looking at a common biological mechanism, or at least a gene that that's common to them both? Then absolutely, I think that's the case. Oh, I think you're on mute.
Thank you for that. I think there are no more questions in the chat box, so I have a question. So, have you explored the role of ZNF84A in the context of neurodevelopment? What time are they expressed, and what could their role be in fetal development, brain development?
Mm-hmm. So I think So with our work, we've really focused here on developing glutamatergic neurons. What we do know is that actually there is an expression of ZNF8048 in neuroprogenitor cells, and so what role ZNF is playing in the neuroprogenitor cells is not entirely clear. What role ZNF is also playing in the developing of other cell types is also not clear. So, I mean, when we looked at the single nuclei data from the fetal brain, the highest expression was in developing excitatory neurons. Then it was actually intermediate progenitors. Then it was developing interneurons and so forth. So there are other cell types, and our work is very much reductionist. So to really truly understand it, we need to probably go either to a more complex system, like an organoid, where we can get more cell types. But yeah, more globally, it'll be really quite interesting to understand what's happening in neurodevelopment.
And also, there are the glial cells and so many other cells that might be expressing it. And
absolutely.
Yeah. So the last question for today's session is there a difference in the intracellular localization during different phases of bipolar disorder? That's
That is a really interesting question, and I don't know. I'm actually not 100% too sure how we would be able to capture that in a cellular system. Although what I would say is we do believe that it is a dynamic protein, and so we've observed a general localization of ZNF804A to change between different cell states. So from a neuroprogenitor to a neuron state, there's differences. And then under different contexts. So if we stimulate the cells with the different stimuli, we believe we do see some changes in the subcellular localization, and it's actually been reported by another group that in response to interferon alpha, for example, in non-neuronal cells, ZNF804A will move from the cytoplasm to the nucleus. So there is evidence that it moves around. Whether this could be linked to different changes or different phases of something like bipolar, I don't know. But that'd be really quite an intriguing question to explore. So thank you very much for that question.
Thank you. So we've come to the end of today's section. Thank you, Doctor, for spending your precious time with us and sharing your valuable thoughts about studying schizophrenia and psychiatric disorder using cellular systems. So to the participants, I would like to tell you that the recording will be available in a couple of days on YouTube, and you could reach out to Dr. Deepak via his email address. And thank you. We will come back to you with the next session in a couple of days. We would also like to receive your feedbacks. The link is on the chat. Thank you so much. Thank you, sir.
Thank you all very much. It's been an absolute pleasure. Thank you. Bye-bye.